Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's class of Histology, the foundation module lecture 4, we are going to discuss the epithelium. But before moving on to the next slide, please subscribe to the channel and press the bell icon. So the learning objective for the today's class are to describe the epithelium, to classify the different types of epithelia, and to discuss the apical cell surface modifications. So from cells to tissues, as the human body develops from single to multicellular, the cells specializes. The body is an interdependent system and malfunctioning of one group of the cell is catastrophic. The cells specialize into types of the tissues that interspersed into the organs. So different types of the tissues which are present in the body, basically four types of the tissue. Most organ contains four types of the tissues. So the first is the epithelium, that forming the covering, the lining of the surfaces. Then is the connective tissue which gives support, and present in examples are the bones, ligaments and fats. Then is the muscle tissue responsible for the movement, and the nervous tissue that is responsible for the control, includes the brain, the nerves and the spinal cord. So basically four types of the tissues are present. The cell organizes to form the tissues and tissues organizes themselves to form the organs. So basically four different types of the cell tissues are present in the body. These are classified as epithelium, the connective tissue, the muscle tissue and the nervous tissue. So today we are going to discuss the epithelium. Epithelium is an avascular tissue. It lines the exterior surfaces of the body. It lines the internal body cavities, the body tubes, forming the parenchyma of the glands and also forming the receptor for the special sense organ. So must remember, it is an avascular. It lines exterior as well as the internal body cavities. And the tubes, body tubes like genitourinary tubes, like gastrointestinal tube, which are connected to the exterior, they are lined by epithelium. And this epithelium is also modified to form the parenchyma of the gland, and this epithelium also forms the receptor for the special sense organs. So what are basically the characteristics of the epithelium? How we classify tissue as epithelial tissue? So there are certain characteristics on which we classify. So what are these characteristics? First, they are closely opposed and adhere to one another by means of the specific cell-to-cell junctions that form the specialized cell junction. So in this diagram, you can see the cells are closely attached, opposed to each other, and they are attached to each other by the specialized type of the junction. So this is one of the criteria for a tissue to be labeled as epithelia. The next is they must exhibit functional and morphologic polarity. This is also very important. In other words, different functions are associated with three distinct morphological surface domains. So in this diagram, again you can see they must have an apical or the free surface, they must have a lateral domain, and they must have a basal domain. So this is the second criteria on which any tissue which is having these criteria, we classify them as epithelium. And this is another diagram in which you can see the different cells. They are closely opposed to each other. They are having the apical surface, the lateral surface and the basal surface. The third criteria for the tissue to be characterized, to be classified as epithelia, is that the properties of each domain are determined by specific lipid and integral protein. And third criteria is that their basal surfaces is attached to underlying basement membrane, that is a non-cellular protein-rich polysaccharide membrane. Here you can see this one is the basement membrane. So these are three criteria. First, cells should be closely opposed to each other. Second, they have the polarity. Polarity means they having the apical surface, they have lateral domain, and they must have the basal domain. And the third characteristic is that they must have a basement membrane. So any tissue which fulfill two out of these three criteria, they are not classified as epithelia, but they are called as epithelioid. Like epithelium, the cells are like epithelia, like the interstitial cells present in the testes. they do not have a free surface. Apical surface is absent. Although the cells are closely opposed to each other and they are also having the basement membrane, but they do not have the apical surface. That's why the cells are given the name of epithelioid. So these are the three basically characteristics. Any tissue which we classify as epithelium, they must have these three characteristics. This is another diagram. The same diagram, you can see the apical surface, the lateral domain, the basal domain, and the basement membrane. So how we classify the tissue, this is also important, and how we name the epithelia. So first name of the tissue indicates number of the layer. That is, either it may be single, that it consists of only one layer of the cells, or it may be stratified, means more than one layer of the cells. Either maybe 2 layers, either maybe 3 layers, 4, 5, or up to 8 layers may be present. So they must there may be more than one layer. So we classify the first criteria to classify, to give name of any epithelium, is either they are simple or they are stratified. Simple mean only one layer of the cell, you can see in this diagram, and stratified means more than one layer. So next is the last name of the tissue describe the shape of the cells. So shape of the cell may be squamous shape, may be cuboidal shape, may be columnar shape. In squamous shape of the cell, width of the cell is greater than height of the cell, and nucleus is present in the center, and it is usually elliptical in shape. In cuboidal cells, both the width of the cell and height of the cells are equal, and the cells are having the central spherically placed nucleus. In case of the columnar cells, the height of the cell is more as compared to the width of the cell, and cell is elongated, nucleus is elongated, and it is basally placed. So these are the three basic types of the cells which are forming the epithelia. The cells may be squamous in shape, cell may be cuboidal in shape, or cell may be the columnar in shape. So naming the epithelium include both the layer, the first, and the shape of the cell, the second name. So that may be stratified cuboidal, that may be simple cuboidal. Then the name may also include any accessory structure. This is also very important. For example, the cells, the epithelium may be having the goblet cells, the epithelium may be ciliated, the epithelium may contain the keratin. Absence of keratin is also very important negative finding. So we have to mention whether the epithelium is keratinized or non-keratinized. Or the special epithelial tissue do not follow the naming convention. These are the pseudo-stratified and the transitional epithelium. So this is the criteria how we name the epithelia. So first we will do the simple squamous epithelium. This is the example, this is the slide showing the simple squamous epithelium. This slide is taken from that of the kidney, and here you can see the renal corpuscle, and this layer of the cell which is forming the parietal layer of the Bowman space is lined by the squamous epithelial cells. So these are the single layer of the flat epithelial cells. You can appreciate the flattened nucleus of these epithelial cells which are present over here. The other example of the single epithelial, simple squamous epithelium is that of endothelium, or that is lining the epithelium lining the hollow viscera, that is the mesothelium. So these are which lines the peritoneal, pleural, and the pericardial cavities. So the simple squamous epithelium means single layer of the cells. These are flat cells. They have the flattened disc-shaped nucleus which is present in the center, and they have the tapering ends. And types of the epithelia, the examples of epithelia may be the endothelium, may be the parietal layer of the Bowman's capsule, may be the mesothelium. So these all are types of the simple squamous epithelium. Next is the slide which you show. This is the surface view of the simple squamous epithelium, and this is again the same slide showing you the renal corpuscle, in which you can see the parietal layer of the renal corpuscle which is lined by simple squamous epithelium. You can appreciate the squamous, the flattened disc-shaped nucleus of these squamous cells which are lining, and the only single layer of the cell are present. So this is the simple squamous epithelium. So what are the function of the simple squamous epithelium? These are the passage of the material by the passive diffusion and filtration or secrete lubricating substances into serosi. They are located in renal corpuscle, in alveoli of the lung, lining of the heart, the blood and the lymphatic vessel, lining of the body cavities like serosa. So simple squamous epithelium, another slide for simple squamous epithelium. Here you can see these are the alveoli of the lung, in which you can see these alveoli are lined by the single layer of the squamous shaped epithelial cells. So this is the simple squamous epithelium which is lining the alveoli of the lung. Again the same slide of the kidney showing the renal corpuscle, and in which you can see the parietal layer of the renal corpuscle, the Bowman space, which is lined by the simple squamous epithelium. And this is the surface view of the simple squamous epithelial cells. When we are looking at the surface, we can see the nucleus is present in the center, and at the periphery you can see the cytoplasm. So this is the surface view of the simple squamous epithelial cells. Now we move on to the simple cuboidal epithelium. The single layer of the cuboidal cube-shaped cells, the height of the cell is equal to the width of the cell, and they are having the central spherical nucleus. Very easy to identify. Thus, you can appreciate the shape of the nucleus and the position of the nucleus, and you can identify the slide. So this is the single layer. So the slide is that of the simple cuboidal epithelium, and function is related to secretion and absorption. Location may be the kidney tubules, the secretory portion of the small glands, ovaries and thyroid follicles. So at all of these places you can see the simple cuboidal epithelial cells. This is another example, the slide taken from the kidney tubule, in which you can see this is the thyroid follicle, which is lined by the simple cuboidal epithelium. This is another thyroid follicle which is lined by the simple cuboidal epithelium, and in the center you can see the colloid which is present. So please do not confuse the colloid with the epithelium. This is the lining of the epithelium, and this is the lining of the epithelium of the follicle, adjacent follicle. Here you can see two follicles are very close to each other, and epithelium of this follicle and epithelium of this follicle, they are so close that they give the false appearance of being stratified. Actually it is not stratified. The follicle are closely placed to each other. So this is a slide for the simple cuboidal epithelium. In this slide, you can see a duct, the longitudinal section of the duct, in which you can see the simple cuboidal epithelial cells are lining the duct. So this is a very simple diagram. Now we move on to the simple columnar epithelium. Simple columnar is a single layer of the column-shaped cells with oval nuclei and which is basally placed. Some cells bear the cilia at their apical surfaces. Some of the type of the epithelia may contain the goblet cell. Function is related to absorption, secretion of mucus, enzymes and other substances. And epithelium may be ciliated type, like propel the mucus, or in reproductive system by the ciliary movement. It helps the ova to move through the fallopian tube. So simple columnar epithelium located may be non-ciliated, lines the digestive tube, the gallbladder, the duct of the small gland. This is very simple. The epithelium is simple, columnar in shape, with goblet cell. This is very important positive finding. You have to write the complete name of the epithelia, and that this epithelium is simple columnar epithelium with goblet cells. It may be ciliated and lines the small bronchi and the uterine tube. So here you can see, you can appreciate the cilia. This is the type of epithelium, the simple columnar line and the apical surface. You can see the cilia. So you have to write the complete name, that is simple columnar ciliated epithelium, which is present. This may be the slide of the uterine tube. Another beautiful slide showing you just simple columnar ciliated epithelium, and in this slide also you can see the simple columnar epithelium with the stereocilia which are present over here. This is another beautiful slide. We are now going to study the pseudo-stratified columnar epithelium. So you should know that in case of the pseudostratified epithelium, actually it is simple epithelium, but height of the cells varies. Some cells are of short length and some cells are taller. Those which are taller, they reach up to the surface, and those which are shorter, they are present at the bases, which gives a false appearance of being stratified, although it is not stratified. So that's why given the name pseudostratified columnar epithelium. It is having a cilia at their apical surfaces, also having the goblet cells which are present. So all the cells originate from the basement membrane, and only tall cells reach to the apical surface. The complete name for this epithelium is pseudostratified columnar ciliated epithelium with goblet cells. It is usually present in the respiratory system. The cells may contain goblet cells and also having the cilia. The nucleus lies at different levels and gives a false impression of being stratified. Function is secretion of the mucus and propulsion of the mucus by the cilia. So as I have told you that it is present in the respiratory system, so its function is to secrete mucus and to propel the mucus. This is another slide in which you can see the pseudostratified epithelium present over here. This was the same previous slide, and in both slides you can appreciate the cilia which are present in these cases. So location of the pseudostratified columnar ciliated epithelium is that it may be the non-ciliated, present in ducts of the male reproductive tubes where the stereocilia are usually present, or in cases of the ducts of the larger glands. And it may be ciliated variety which is lining the trachea and most of the upper respiratory tract. So in this type of epithelium you can see here is the basement membrane, and next is the cells which are basally located. They are having the short length, and these are the tall cells which reach up to the surface, and among these tall cells you can see the goblet cells are also present, and at the surface you can appreciate the cilia are also present. Even you can appreciate the darker line which shows the basal bodies from which the cilia originate. So these are the basal body which are present over here. Now we move on to the stratified epithelia. This is also very important. The stratified epithelia contain two or more than two layers. They regenerate from below. Major role is protection. They are named according to the shape of the cells which are present at the apical surface. This is very important point you have to understand, because in case of the stratified epithelia there may be number of the layers, maybe up to 8 layers may be present in some cases, and the shape of the cells vary. In sometimes at the bases you can see the cuboidal cells, at some places you can see the polyhedral. So how to name such epithelia? So we name such epithelia on the shape of the cells which are present at the apices. This is very important. So in this slide you can see this is the area where you can see the basement membrane. You can see with my cursor, this is the basement membrane, and next is the from here up till here. I can show you with the pen that from here, this point to this point, you can see this is the whole thickness of the epithelium, and here you can see this one is the basement membrane which I am showing you. This is the basement membrane, and these are the basal layer. These are the basal layer which are present, different layers, and here you can see these nuclei which you can see. These nuclei are squamous shape. Different nuclei, you can see these are the squamous shaped nuclei. So how we name this epithelium? The first criteria is that this epithelium is stratified, means it is stratified. Then different types of the cells are present in this epithelia, but the shape of the surface cells are squamous, which is evident by their nuclei which are present, which are squamous in shape. That is why we call the epithelium as the stratified squamous epithelium. So this is another slide for the stratified squamous epithelium, and in this slide you can see this is the basement membrane from here. Then you can see a number of the layer from here up till here. You can see the epithelium which is present from here to here. This whole is the thickness of epithelium, and this shows you that it is stratified. Next, you can see the shape of the cells. You can appreciate the shape of the cell. These are the squamous in shape. You can see the squamous shape of the cell which are present. Next important thing which you must appreciate in this slide is this area. You can see over here this area. This is the keratin. This is very important and very important finding, whether it is present in stratified squamous epithelium or if it is absent. You have to write the name that it is the stratified squamous epithelium with keratin, or you can see the stratified squamous keratinized epithelium. And if it is not present, because you know that the keratin is present only in the stratified squamous epithelium, so if stratified squamous epithelium do not contain keratin, you have to write that it is non-keratinized type. And if it contains keratin, so you have to write that it is keratinized. So this is important positive finding you have to mention while you are writing the stratified squamous epithelium. So, stratified squamous epithelium is a specific type, may be keratinized, may be non-keratinized. Keratin is the contain the protective keratin. Coat surface cells are dead and full of the keratin. Non-keratinized form moist lining of the body openings, like it may be present in the esophagus and oral cavity, etc. So, protection, it's very important. It protects underlying tissue in areas subject to abrasion, especially the keratinized type of epithelium. It is present. It is the epithelium of the skin, and it is a rough epithelium. It gives a protection to the epithelium, so it is very important. Keratin is very important. Location is the keratinized epithelium is present in epidermis, and non-keratinized epithelium forms the lining of the esophagus, the mouth, and the vagina. So now we move on to the transitional epithelium. Again, you can see in this slide, first we will mark the basement membrane. Here you can see this is the basement membrane. You can see this all is the basement membrane. Next we move on to the number of the layers. So you can see from here to up to the surface, you can see this is whole thickness of the epithelium. So it means the epithelium is stratified. Our first naming criteria that it is stratified. Next, the surface cells, you can appreciate the surface cells. You can see the surface cell. So these are typical dome shaped cells. They have the typical domes which are present at the surface of these cells. They are the typically dome shaped cells which are present, in which in also over here you can see they are typically dome shaped cells. And also important finding, some of the cells may be binucleated. So in this you can also appreciate binucleated cell. So these are the different criteria. First, the epithelium is stratified, then the typical special type of the cells which are present at the apices, these are the dome shaped cells. So that's why, and this epithelium is considered as a transition between stratified cuboidal and the stratified squamous epithelium. So it is given the name of the transitional epithelium. So very important, the transitional epithelium. So, transitional epithelium, basal cells usually cuboidal or columnar. Superficial cells are typically dome-shaped. Here you can again see these are the dome-shaped cells which are present, and function is that stretches and permits distension of urinary bladder. This type of epithelium is present in urinary tract. It is also given the name of the urothelium, which stretches and permits the distension of the urinary bladder. And location is, as I have told you, ureter, urinary bladder, and part of the urethra. So next is the stratified cuboid. So stratified cuboidal epithelium basically present in male urethra, in duct of glands like the sweat glands, etc. So here first I will mark the basement membrane over here, so you can see all around this is the basement membrane. This is one of the duct, some of the duct of the gland. Next important thing you must know that it is composed of the two layer. One layer is this, and the next layer I will mark is with another color. So this is the next layer. So this is the shape of the cell. Sorry. This is the shape of the cell, which is cuboidal in nature. And here you can see the nucleus of the cell. You can appreciate the nucleus which is present over here. So in this slide you can see first this is the basement membrane. Second, you can see two layers of the cells are present, the one at the bases and other at the surface. And third important point is that surface cells are typically cuboidal in nature. They have this spherical nucleus which is present. So as our naming criteria, we look at the shape of the cell which are facing the apical surface. That's why this epithelium is given the name of stratified cuboidal epithelium. Next is a stratified columnar epithelium. So in this slide also you can see two layers present. First I will mark the basement membrane. So this is the basement membrane. Next I will mark the basal layer. So from here to you can see this is the first layer which is present over here. This is the basal layer, and next you can see with another color that this is the another layer which are present over here, and you can see these cells are tall columnar cells. So according to our classification criteria, the first layer, this one is the first layer and this one is the second layer. So this epithelium is stratified. And next important point according to naming classification is that surface cells are tall columnar. That's why the epithelium is given the name of stratified columnar epithelium. This epithelium may be present in epiglottis, may be present in conjunctiva of the eye. This may be present in the pharynx and the mucous membrane as well. So these are the different locations: the simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium with goblet cell. Here you can see the stratified squamous non-keratinized type of epithelium. Here you can see the stratified cuboidal epithelium, and here you can see the stratified columnar epithelium. So important next topic which we are going to discuss in today's class is the cell polarity, which I have already mentioned you that the polarity means that these cells have an apical or free surface, they have a lateral surface, and they must have a basal surface. And this is very important because they have different proteins which are present. The apical surface have different proteins. These are the channel proteins. These may contain some enzymes as well. Basal surfaces have different proteins, and the basal surface have different protein, and lateral surface also have different protein. So this is very important, the cell polarity. So apical domain, lateral domain, and the basal domain. What are the specialization of the free surface? That is the apical surface. There may be microvilli present on apical surface, there may be cilia and flagella present on apical surface, or there may be the stereocilia present on apical surface. So first we will discuss the microvilli. These microvilli are the finger-like extension derived from the cell surface, and function is to increase the surface area for absorption, cell movement, and the phagocytosis. Localization: cells specialize for absorption, having the microvilli like in intestinal epithelium. They are very organized microvilli and given the name of the striated bladder, and in case of proximal tubule of the kidneys, again they are very organized, well formed microvilli. They are given the name of the brush bladder. May be present in the gallbladder epithelium as well. So these are the three different locations you can see. Here you can see the blunt microvilli over here in this surface. In this slide you can also see this is the electron micrographic picture from different epithelia, and in this type you can see the epithelium also the blunt and not well organized, but in this case you can see very well organized microvilli are present. So this is the epithelium of the GIT. Again you can see, so what is the structure of microvilli? So in this diagram you can see the core of the microvilli is formed by the actin filament. This is very important. core of the microvillus is formed by actin filament, and at the tip are positive, and these are anchored by another protein called the villi. In addition, there are three other proteins which cross-link these actin filaments. These proteins are aspin, the fibrin, and the facin. So these three proteins which are present, they cross-link the actin filaments, and these actin filaments are forming the core of the microvillus. Another protein called myosin 1 protein is present, and this myosin 1 protein bind the actin filaments to the plasma membrane to help to anchor these filaments. At the basis of the actin, at the basis of the microvilli, these actin filaments, they cross-link with each other and form the terminal web. And in this terminal web, you can see special type of the protein which is present called the spectrin protein, which is responsible for cross-linking, and also myosin 2 is present. And at certain locations, the tropomyosin is also present. The presence of tropomyosin and myosin gives an indication that these are contractile in nature. So again, I repeat, the core is formed by the actin filament, and it is anchored at the tip are the positive end by villi. Three other types of the proteins which are present which cross-link these actin filaments are the aspin, the fibrin, and the facin. The myosin 1 protein is present which help to these actin filaments to attach to anchor to the cell membrane, the plasma membrane. This is the terminal web where the cross-linking of these actin filaments are present, and to help cross-linking we have the myosin 2 filament, and tropomyosin is present, and also this terminal web is attached to plasma membrane with the help of the spectrin. This is the electron micrographic picture of the again the microvilli and the stereocilia as well. The stereocilia are actually unusually long microvilli, and recently the histologist gives the name of stereocilia as a stereovilli as well, because they have the same character as that of the same internal organization as that of the microvilli. They do not resemble to the cilia, so that's why they are called the stereocilia. Actually they are the stereovilli. So in this diagram also you can see the microvillus, the actin filaments, they are cross-linked with the help of the fibrin and the aspin and the other filaments. At the tip you can see villi which is present, and at corners you can see the myosin 1 is present which is attaching these filaments linking to the cell membrane. This is the area of the terminal web where you can see the myosin 2 and the tropomyosin which cross-link these actin filaments, and also the linkage to the cell membrane where you can see the spectrin is present. So this is the structure of the cilia. You can differentiate between two. The core of the microvillus is formed by actin, while in the cilia you can see the triplet of doublets of 9 with the 2 microtubules in the center. This is the structure of the cilium. We will do it in detail now. So microvilli, quickly we will go through the detail. The finger-like projections 1 micrometer in length, 0.1 micrometer in diameter, can be seen with electron microscopy. Contain bundle of straight parallel filament 20 to 30 actin filaments. Actin filament extend 0.5 micrometer down into apical cytoplasm, enclosed in an extension of the plasma membrane. So this is the slide of the GIT, in which you can see the simple columnar epithelium with goblet cell and also large number of the microvilli which are present over here. And these microvilli over here function as for the function of the absorption. They are responsible for absorption of the material which is present in the lumen of intestine. The striated border contain enzyme. This is also very important MCQ. In small intestine, the striated border of the microvilli contains the enzyme that is sucrase, maltase, lactase, lipase, and aminopeptidases, and they are involved in the terminal digestion of protein, carbohydrates, and lipids. And another important point you must remember that these enzymes are present only in small intestine, not in the large intestine. So now we move on to the cilia. A little bit about cilia. They are unusually long and irregular microvilli, 8 micrometer in diameter, have internal actin filaments bundled cross-linked by the fimbrial structure instead of myosin. It has a protein called asrine. This is a point which is differing from the microvilli. Here the instead of myosin 1 we have the asrine protein to attach this actin to the plasma membrane. Also absence of the villi at the tip. This is villi, and in case of the microvilli, villi is present. In case of stereocilia, this villi is absent. This is a mistake in spelling. Please note it. Then these are non-motile. We know that the cilia are motile, but these are non-motile. And basically no basal bodies are present because cilia arises from basal body. So there are no basal body, and function is to increase the cell surface area for absorption. Location is ductus epididymis, ductus deferens, and sensory hair cells of the ear. So this is the beautiful slide of the cilia, the electron micrographic picture. You can see different stereocilia which are present. Now we move on to cilia, the kinocilia and the flagella. So the cilia have the eyelash or hair-like structure. They are motile. They are larger than microvilli, 5 to 10 micrometer long and 0.2 micrometer in diameter, have complex internal structure. 250 or more cilia in each cells are present, and they are arranged in parallel rows. Location is epithelial cells of the respiratory tract, epithelial cells of uterine tube, and vestibular hair cells of ear. And the ciliary movement is constant in a direction in living cells. Cilia beat in a rhythmical wave-like manner and to transport the ovum towards the uterus. So this is the structure of the cilia. We can see the doublets of the microtubule which are formed by the microtubules doublets having the A and the B tubules, and in the center you can see the two separate microtubules. The cilia having the hair-like structure. No internal structure is detected at with the light microscopy. You cannot appreciate any internal structure with light microscopy. At base of each cilia, a dense granule basal body is seen with electron microscopy. They have a complex internal organization. The arrangement of microtubules called axoneme 9+2 microtubule complex. We will do it in detail. Each cilium is covered by extension of plasma lemma having tip, a cylindrical shaft, and a basal body. So this is the structure of the cilia. You can see the doublets of the 9+2 doublets which are present in the periphery, and in the center you can see the two separate microtubules. Here you can see the A microtubule is complete having 13 protofilaments, and the B microtubule share the part of A with it having 10 tubulin molecules in it. Another important point is that they have a pair of the dynein arms which requires the energy of the ATP for movement, and this is responsible for movement of the cilia. Then they have another protein, a nexine protein, which is important. This protein bind the B microtubule of one doublet to the A microtubule of the another doublet. Then you can see in the center you can see the central sheath projection is present, which is surrounding the central pair of the microtubule, and to this central sheath projection the radial spoke of each is attached. So this is the whole structure of the cilia, you can see, and this is the shaft and the tip of the cilia, and this is the transitional zone, and here you can see the basal body from which the cilia arises. So basal body have triplet of 9 structure, and out of these triplet the C protofilament C tubule is present in the transitional zone, but only A and B are forming the structure of the cilia, the basal body having triplet and cilia having doublet. Another important point is that basal body do not have a central 2 microtubules, while the cilia having the 2 microtubules in the center, and these 2 microtubules arises in this transitional area de novo. So this is the difference between this, and basal bodies basically is the region which is responsible for formation of the cilia, and this region is called the MTOC, that is the microtubule organizing center. So Nexin links each attach microtubule A to B of the doublet, and Nexin links are composed of elastic material called Nexin. So this is the Nexin, and this is responsible for the recovery stroke. So see, these are the cilia of the tracheal epithelial cells. Then the basal body, as I have told you, the triplets of 9 and no central microtubules which are present in it. Then the mechanism of the ciliary movement is, old concept was that ciliary movement is based on contraction of the microtubule, but the newer concept is, which is now accepted, is that the cilium bends along the axis by the type of sliding microtubule mechanism between microtubules. So it is similar to that seen between myofilaments in the striated muscle fibers. Then the dynein arms, as I have told you, these are extremely large proteins. The dynein arms form temporary cross bridges between microtubule A of one doublet with that B of the adjacent. So during sliding, they undergo a cyclic break and reattachment. Formation of the cross bridges is ATP dependent. So dynein walking model is that dynein appears to walk along the adjacent doublet. Experiment is that proteolytic enzyme digests the Nexin link and the radial links. So addition of the ATP produces a sliding movement of the bridge along the B tubule of the adjacent doublets. Doublets move relative to each other, powered by motor activity of the axonal dynein. So radial linkers convert the sliding of microtubules into bending of the cilia. So ciliary movements requires ATP and calcium, and this is again very important MCQ as well. So structure of the microtubule is that microtubules consist of the 13 protofilaments. The protofilaments are composed of the tubulin subunits are dimer, and they have the tubulin A and B, tubulin B dimers, tubulin alpha or beta dimers. The flagella next is the same internal structure with cilia, that is axoneme 9+2, long whip-like protrusion whose undulation drive a cell through the fluid medium. The eukaryotic flagella are larger than the cilia. The bacterial flagella are smaller and different mechanism of action. So flagella having longer than the cilia, 100 to 200 micrometer in diameter, and there are different types of the movements. Undulating wave type movements are less in number, 1 or 2 in a single cell. There may be less in number. The mammalian sperms contain 9 additional dense fiber around the axoneme. This is 9 plus 9 plus 2. This is the mammalian sperm internal structure. So these are the references for today's class, that is Basic Histology by the Lekus and Siddiq, the Histology text and atlas by Janquera, and Histology text and atlas by H. Ross. Thank you very much for your attention for today's class. Allah Hafiz till the next class.